infections that target blood and lymphatic system. So the lymphatic and cardiovascular systems are the two major systems responsible for moving fluids around our bodies. They work together to make sure that our tissues get what they need, all the nutrients, for example, that they need, but also they get rid of what they don't need, waste of the metabolic processes. So pathogens that reach these systems and produce what we call a systemic infection, they may cause in many instances what we call septic shock. And because this is something that can be caused by several pathogens that we're going to mention in this chapter, let's start by defining that septic shock. So septic shock is a life-threatening condition caused by severe, localized, or system-wide infection that requires immediate medical attention. And symptoms may include, for example, low blood pressure, chills, problems breathing, mental confusion, disorientation. It will require emergency treatment, and that may include, for example, supplemental oxygen, IV fluids, antibiotics, other medications could be used there too. So how pathogens may lead to this stage of septic shock? Well, it is most commonly caused by bacteria, but also it could be caused by fungi, by viruses, or parasites. So it's not exclusive for bacteria. Most cases of septic shock are caused by gram-positive bacteria, followed by endotoxin producing gram-negative bacteria. And this figure here that you see over here is basically summarizing, it's showing you how the endotoxin released by gram-negative bacteria may lead to septic shock. And this includes, for example, a massive activation of macrophages that results in a cytokine storm, and that leads to a shock impaired oxygen exchange. Or, for example, it can also cause complement activation, and that will lead to lung tissue damage. Or may result in disseminated intravascular coagulation, DIC, disseminated intravascular coagulation, that leads to an hemorrhagic process that will now produce low blood pressure and organ failure. So let's just start with the bacteria that is a gram-negative bacteria that has that endotoxin that may lead to septic shock. And the bacterium that we are analyzing here is called Yersinia pestis. That's the scientific name for that bacterium. And that bacterium is associated to a terrible disease. So there is DNA evidence that was recently published that Yersinia pestis infected humans 5,000 years ago. We're talking about the Bronze Age, okay? But genetic changes that made it highly virulent, they did not occur until about 4,000 years ago. And then the Black Death or the pestilence or the plague, this was the deadliest pandemic recorded in human history. The Black Death resulted in death of about 200 million people in Eurasia and North Africa, peaking in Europe in the mid 14th century. So the natural reservoir for this bacterium is believed to be principally the marmot or the prairie dogs. This is, we believe that that is the natural reservoir of that bacterium. And several species of rodents, they could be also, important reservoirs for that bacteria. So the main vector is considered infected fleas. You have a picture here of those fleas. So in the case of plague outbreak, many rodents die after being bitten by infected fleas, and then the fleas are forced to search for other sources of food, like, for example, human blood. And then now humans get infected, and we are considered terminal hosts. There are three forms of plague. We have what is called pneumonic plague. We also have the septicemic plague. And finally, we have the bubonic plague. The pneumonic form may occur following an initial bubonic or septicemic plague infection. And it may also result, from breathing in airborne droplets. So there are different ways in which this bacterium can be transmitted. It can be a zoonosis by transmission through this vector, the flea, infected fleas, or it could be airborne and actually, through droplets, directly through droplets. That's the main transmission in the pneumonic plague. And these are the ways in which the bacteria could be transmitted. So when that bacteria gets into the body, the bacteria brings important virulence factors, like, for example, the Yersinia outer proteins, this right here, JOPs. These are proteins that the bacterium produces, and these proteins are injected into the cytoplasm of host cells. And then the injected JOPs help to limit phagocytosis. And in addition to that, the bacterium also produces a capsule that further, helps on preventing phagocytosis. Those are important virulence factors. Of course, this is a gram-negative bacterium, and in addition to that, because of the outer membrane and the lipid A that is present in that outer membrane, that lipid A is an important endotoxin that can cause septicemic plague. Thousands of cases of plague occur every year around the world. And with proper antibiotic treatment, the prognosis of this infection is now much better. The plague is now commonly found in sub-Saharan Africa and Madagascar. And in the United States, only few cases may happen. But again, if the individual is treated quickly with antibiotics, the infection should be cleared very easily. The treatment of this infection is basically, using antibiotics. Pneumonic plague is the most serious one, right? The one where the bacterium reaches the lungs. And this is the most aggressive infection and has the highest mortality rate, approximately 100%. And that requires early treatment. Antibiotics must be given within 24 hours of the first symptoms to reduce the risk of death. And which antibiotics can be used? Well, several antibiotics are effective against Yersinia pestis. Like, for example, streptomycin can be used, gentamicin, tetracycline. Chloramphenicol can be used, but remember that chloramphenicol can have some serious adverse events. And they can all kill the bacterium and stop the infection. We don't have any current vaccine to prevent that infection. There are some vaccines that are under study, but not a single vaccine has been approved to be used in humans yet. I forgot to mention that tetracycline is a common antibiotic that can be used to treat Yersinia pestis infection. Let's now go and talk about a virus that targets the lymphoid system. And the virus that we're talking about here is the Epstein-Barr virus, or EBV. This is a type of herpes virus. Another name for the Epstein-Barr virus is herpes, human herpes virus type 4. And the disease that this virus is associated to is called infectious mononucleosis, or also the kissing disease. How this infection happens? Well, usually this is a disease that is going to be transmitted through oral pharyngeal secretions, saliva, for example. Like if you shared on a spoon or a glass, and there is saliva there, and if you have the virus and now you share that glass, that spoon with another person, that person may contract the infection. Or by kissing, that could be a route of transmission. And that is the main reason why it is called the kissing disease. When that happens, epithelial cells of the oral pharyngeal area, they become the portal of entry for the Epstein-Barr virus infection. And the virus is transmitted primarily by this repeated contact with oral pharyngeal secretions. And most cases of infectious mononucleosis, they are mild or moderate, but then symptoms of the infection, infectious mononucleosis, they can include fatigue, fever, rash, swollen glands. And following this primary infection, Epstein-Barr virus can persist lifelong in the host by selectively infecting latent state memory B cells. So the B cells, those cells that are in charge of producing antibodies, they are the target for this Epstein-Barr virus. And there the virus can do a latent infection. And that latent infection of B cells has been associated with a particular type of cancer that is called Burkitt's lymphoma. Now, how do we treat this infection? Well, we don't have any selective antiviral for this virus, and supportive therapy, including bed rest and analgesics, is the primary form of treatment for this infection. We have no vaccine to prevent EBV infection, okay? Here we are now with another virus, and this is a virus that produces a zoonotic disease because it's transmitted to humans by animals. Yellow fever is an acute viral hemorrhagic disease that is transmitted by infected mosquitoes. And the yellow name of that virus refers to the jaundice that affects some patients, because the virus may affect the liver too. So after transmission from a mosquito, the virus replicates in the lymph nodes and then infect dendritic cells. Dendritic cells is a very important, cell that this virus infects. And from there, they reach the liver to infect hepatocytes, which leads to the release of cytokines. And then now fatality, death may occur as a result of a cytokine storm that leads to shock and to multiple organ failure. I mentioned to you that this is a zoonosis because there is a vector that transmits that virus to humans, and that vector is the Aedes aegypti. Is a major vector that is not only transmitting yellow fever, it may also transmit other diseases like dengue fever, Zika, chikungunya. And we have no proven antiviral therapy for this. Basically, only supportive care can be used for the patient that is developing a serious disease. However, we have a very highly effective attenuated viral vaccine. And this is a vaccine that you get only one shot and provides lifelong immunity for the rest of your life. So since we are talking about viruses that are transmitted by this mosquito, let's now talk about another viral infection that is transmitted by this same mosquito. And now we are talking about dengue viruses that produce dengue fever or hemorrhagic dengue fever. So in the case of dengue fever, once again transmitted by mosquitoes, there are different species that may transmit dengue virus. Once the virus is introduced in the bloodstream, once inside the skin, the virus binds to Langerhans cells. This is a population of dendritic cells in the skin. And then the virus enters the cells, start to replicate in those cells, and now produces new viral progeny that are released from these cells. And those viruses, they get into the bloodstream and they start to infect white blood cells, like for example, monocytes and macrophages. The classic dengue fever or break-bone fever, that's another name for that disease, is characterized by an acute onset of high fever, and then the symptoms include frontal headache, retroorbital pain, that's in the back of the eye, hemorrhagic manifestation, RASH, and low white blood cell count. That's very characteristic of this infection. And some patients with dengue fever, they may develop what we call dengue hemorrhagic fever, or DHF, right here. This is a severe and sometimes fatal form of the disease. And the mechanism by which patients develop this dengue hemorrhagic fever is called antibody-dependent enhancement. Let me write that down. Antibody-dependent enhancement, or the acronym that is ADE. We're going to talk about this mechanism during our lecture meeting, and you will see there how this mechanism allows dengue virus to now produce a more severe and serious infection. We do not have, selective antivirals to treat the infection by dengue virus. However, vaccines have been developed, and this vaccine, Dengvaxia, has been developed, and it was approved recently by the FDA. And one of the concerns with this vaccine could be increased risk of severe disease through this ADE mechanism. And that happens especially in children who receive the vaccine and have not been previously exposed to dengue virus. So the vaccine is effective, but it has to be given to people that were previously exposed to dengue. Those are the ones in which the vaccine is going to be safe. And I mentioned here that there are different serotypes of dengue. And when we meet, during lecture, I will explain how, because of these different serotypes and this mechanism, in some patients, the dengue hemorrhagic fever can be produced. Let's now talk about another virus that infects cells in the that are circulating in the bloodstream, but also some other cells can be infected by this virus. And this is a virus that probably is very familiar to you, Ebola virus, that produces the Ebola virus disease. And this is a virus that produces emerging hemorrhagic fever. And in the same family of Ebola virus disease, you also have the Marburg virus disease. They both belong to the same viral family, the Filoviridae, and they both can cause these emerging hemorrhagic fevers. So Ebola, again, is a viral hemorrhagic fever of humans and other primates, non-human primates, that can be caused by these Ebola viruses. The virus spreads through direct contact with body fluids, like, for example, blood of infected humans or other animals, and then a spread could occur from contact with items that have been recently contaminated with infectious body fluids. In terms of the reservoir for this virus, it has been proposed that fruit bats could be the main reservoir. And then now those animals may transmit the virus to humans, and then humans are what we consider a terminal host. And the word terminal there, fits very well, because depending on the strain of the virus that is causing the infection, mortality rate could be really, really high. The case fatality rate goes between 25 and 90%. So the Ebola virus, in terms of the pathogenesis, the Ebola virus can replicate efficiently in many cells. And that includes mononuclear cells, monocytes, phagocytes, but macrophages, dendritic cells. It may also infect hepatocytes, fibroblasts. And then that viral replication triggers a high level of inflammatory cytokines, producing a cytokine storm that leads to septic state. So endothelial cells that are also infected by the virus, they start to break down. And remember, the endothelial cells are the cells that are part of the blood vessels. When the virus replicates there, those cells are lysed, destroyed. And now that leads to blood vessel injury. And the widespread bleeding that occurs in affected people, that causes swelling and shock due to the loss of blood volume. I mentioned already that fruit bats are the suspected reservoir for this virus. And then, regarding treatment and prevention, there are currently 2 treatments that is not mentioned here in the slide, but I'm going to write it down now. There are 2 treatments that were recently approved by the FDA in 2020. And the treatment is with Immacep. This is a combination of 3 monoclonal antibodies. And then the other one that was recently developed is a treatment with a monoclonal antibody that is called Ivanka. In addition to that, patients have to be kept hydrated, oxygen levels maintained, a good supportive care during that treatment. But even though that supportive care is happening, still a lot of people will die as a result of that infection, because the case fatality rate for this virus is very high. Recently, a vaccine was developed, this vaccine that I'm mentioning here, Ervebo, and it was approved in the United States in 2019, and it appears to be fully effective 10 days after being given, and the efficacy is very high. And it was a study in the recent outbreak that happened in West Africa between 2014 and 2016. So the final pathogen that we are discussing in the context of infections of the blood and lymphatic system is malaria. And I am not going to discuss malaria because team number 4 already, put together that presentation, and the presentation has been published on Blackboard. So you can look at that presentation from team 4 that is walking you through this important infectious disease, one of the most prevalent infections worldwide. So this is the end of this chapter. The next topic that we are discussing will be infections of the nervous system and infections of the urinary tract and reproductive tract, sexually transmitted infections.